Okay. Am I back live? I hope. Trying to rearrange my desktop a little bit so I can hopefully see all the different things I need to see here. Okay, I'm watching the chat on the YouTube channel and keeping track of here. All right. Guillermo's done. Stream ended. Starting again. It's just a nice. At least the first 7 minutes. Okay, I'm not seeing myself back. Starting again, video, and okay. All right, I think I'm back. Are we all back? That's a real pain. Trying to. I just need another monitor, and it'd be quite a bit easier to keep track of all these things. Okay, good. At least two of you are back. Melissa and Oscar, and hopefully everybody. Guillermo was another one who chimed in at some point. You're not getting it. Okay, just needed to refresh the page. Hopefully. Okay. So, feel a little awkward here since it's monologuing a little bit instead of discussing. Okay, thanks Oscar for the tip to press F5 to refresh. Okay, so I brought up Mark Rober's 4-step design process, and this is a little different than we can't define science real easily because it's very diverse. So engineering is a type of science, but with engineering there's, you're working towards a measurable goal of some sort, and you're trying to optimize a design oftentimes. At least that's what this video was about. Did anything stand out? How did we do on predicting which variables would be important? What did we get? What did we miss that they included? Okay. You're on your phone, Guillermo. So is Guillermo still not seeing it? Okay, yeah, angle, that 20-degree angle was interesting. It was interesting that it pushed down and then it made its own ramp and it had the momentum to go up. So the speed was important. They didn't adjust the speed since they had a thrower, but the angle was really important. The shape of the rock they controlled for, so that was. Can we just check and see if Guillermo's with us yet? I'm a little hesitant to keep going too far. Guillermo, are you seeing hearing again? Yes. It would be odd to be able to see the comments coming in but not get a live feed. So being on the phone, Melissa, okay, so you're on your phone, but are you seeing and hearing just fine? Really? Guillermo equals Amy. Did not know that. Okay, well, just looking to see. Amy, Melissa, do you see, do you hear? If not, let me just, I've got people in the other section. Okay. It was. Okay. Sorry. Great. Everybody's back. Sorry to be answering a text there. Has to do with the tests. Okay, so yes, we were saying that we got some things, we didn't get all things. The angle was a factor that we didn't get, and maybe spin as well. All right. Now, as I said, there are a lot of processes. Science is very messy typically. This example was actually a lot cleaner than most because there was a very specific goal. There was already a lot known, right? People have been skipping rocks. Mark Rober had experience skipping rocks. He knew, okay, he knew that there was variation. Some people were better than others. He had a sense already of, you know, some of the factors, and then he did the research. Now, the research often can be quite lengthy. If you're going to science journals, and let's just go to plusone.org. This is plusone is nice as a science journal because it's open source. And probably if we search for rock skipping here, we're not going to find anything, unfortunately. But plusone hasn't been around for very long, and you can see that it's divided: biology, medicine, computational biology, the good, the bad, the recovery, and assisted migration, trends in corneal transplantation. I don't know how they're getting rock skipping out of these things, but the word rock and skipping must be in these things. So let me look for something. Let's do something like diabetes, which of course there's going to be a lot of science around diabetes. So let's just take a look for the, you know, doing your research part of things. The problem a little bit is that diabetes is too broad a term, too broad, and you're going to get too many results, right? People have been studying diabetes for quite some time, so where do you even start? It's just going to be a lot of background research. You're going to have to do a lot of reading, a lot of study. So in the case of something like diabetes, starting with physiology textbook is good, and reading some of the older papers, and then you're going to have to work your way to a little particular branch, right? Diabetes is this huge thing, and you're going to have to narrow it down to, well, I'm interested in the insulin response of muscle cells, and that's much narrower. So if you start looking at insulin receptivity on muscle cells, that would be a much narrower search. And this, you can't do search just at plusone. This is just one journal. You're going to have to look across many journals, and there are tools to help you do that. So you end up doing a lot of research, and that really can take so much time because you're just trying to get to the background knowledge so that you can then push forward into some new area. And with my own research on horned lizards, just as an example, I did several years of research, and I took my data and all the things I had been learning, and I presented it to my graduate committee. So a group of 5 professors who were my committee as I pursued my PhD. And after 3 years of research and study and everything, they weren't convinced that I had enough background knowledge yet to know about desert ecology and horned lizards and this particular species. And I had one professor on my committee in particular who was pretty much just a walking encyclopedia of research articles. So I would say something, and then he'd say, well, doesn't that contradict what Smith and Slobodkin said in 1974? And I'm thinking to myself, I have no idea, you know, what was said in 1974 by Smith and Slobodkin, so I have no idea if I contradict them or not. So I'd have to make a note to myself to go check on that paper. And then I'd say something else, and he'd say, yeah, but hasn't that already been done by Hairston in 1992? Or I don't know, has it? So the background research can really be difficult. And I know this is hard to see, but let me just pull up any one of these studies. Higher levels of physical activity are independently associated with a lower incidence of diabetic retinopathy in Japanese patients with type 2 diabetes, a prospective cohort study. Okay, so these titles are going to be long and a little difficult, but what I wanted to show you, there's the abstract, but I wanted to get into the actual paper. And I will try to enlarge this. I know some of you are on phones and won't see this very well at all. But first, a few things about science nowadays. One, it's completely international, so studies are coming from all over the world. These are Japanese authors. The study was in Japan. Two, English has won out as the international language. So if you speak English, that's very helpful. And even if you don't speak English, it's helpful if you can get your paper translated into English, because that has become, you know, it used to be French. Not that long ago, French was considered the international language, and if you were going to do science, you had to learn French. Well, now it's English. So, but what I wanted to show you is that any statement you make, the prevalence of type 2 diabetes mellitus has significantly increased worldwide, which has in turn increased the burdens on individuals and healthcare systems. That's a pretty generic phrase. They're just saying, well, diabetes rates have sure gone up, and that's kind of like, well, yeah, everybody knows that. Well, you can't just say that. You have to cite somebody. So they chose to cite Fenwick, Rees, Pesadovs, Durrani, Kawasaki, and Wong, and others. Their paper, Social and Emotional Impact of Diabetic Retinopathy, a Review. So they're citing a review paper, and that review paper basically is looking at the literature of the past 10 years and summarizing it, the literature on one subject. So in doing your research, one of the things you find is papers just like this one, where it's a review of a whole bunch of other papers, and that's helpful. But still, if all you're relying on is a review of all these other papers, you may miss some details in those other papers. Okay, so they say that. Then they have another phrase. One such burden is the increased prevalence of chronic complications, such as diabetic retinopathy. In other words, diabetes causes problems with the retina. Diabetes is the leading cause of blindness now. So DR, diabetes retinopathy, has an insidious onset and is a major cause of vision loss in patients aged 20 to 64 years. So it's a major cause of blindness for people 20 to 64 years old. Again, you can't just say that. You have to back it up with something. So they cite another paper, right? Prevalence and causes of visual impairment and blindness among 9,980 Scandinavian adults. It's a big Copenhagen eye study. Great. So this is part of your research, is just knowing kind of who said what, when, so you can back up pretty much any statement. So I'll just do, this is just the introduction to this paper, right? Thus, effective diagnostic methods and therapeutic tools are needed to prevent diabetes retinopathy in patients with diabetes, identification of clinical features in patients with diabetes that can predict the development and progression of DR is crucial. Suggested risk factors include a long history of diabetes, elevated blood glucose levels, increased blood pressure, and impaired kidney function. So what are they saying? They're saying we should, you know, because this is a big problem, it would be helpful if we could diagnose it well. And here are some of the risk factors that we want to consider. Having diabetes for a long time, having elevated blood glucose levels for long periods of time, increased blood pressure, and impaired kidney function. Those are all risk factors for this diabetes retinopathy. And they cite 4 papers now. It's 3 to 7. Anyhow, that's just the research part of things. And they're just backing it up. And now they're describing what their actual study is in the materials and methods. So the introduction in a scientific paper, in a journal article, gives the background. And usually at the end of the introduction, they will transition into what is their hypothesis or what is their study about. They'll set up a problem. In this case, it says, however, few large-scale prospective studies have evaluated the association between PA, which is what? Where's PA? Physical activity. Between physical activity and newly developed diabetes retinopathy in patients with type 2 diabetes. We studied data from a large-scale, single-center registry of Japanese patients with type 2 diabetes to elucidate the prospective relationship between baseline physical activity levels and the development of diabetes retinopathy over a 2-year follow-up period. So that's it. That's what they've got. Their question is, what's the association for people who already have diabetes? What's the association between physical activity and diabetes retinopathy? And they're saying this is a question worth asking because there isn't a big study looking directly at this yet. So people have looked at physical activity with other aspects of diabetes, which they talked about a little bit, but they haven't looked specifically at this. So then they describe their materials and methods. You know, how did they choose who was going to be in their study? And if you think about that, go back to Mark Rober and the rocks. Oh, sorry, I wasn't watching the chat there. Oscar had some questions earlier. Go back to Mark Rober and the rocks. They wanted to control as many variables as possible, so they made their own fake rocks, right, out of clay, so that they would have consistency there. So if you're wanting to do some study with diabetes, if you don't narrow down who's going to be in your study, then all sorts of other factors might start coming into play. If you have very old and very young mixed together, that might make for messier data, right? If you have very skinny and very overweight, that might mess up the data. So you want to kind of narrow it down a little bit, put some boxes around it so that you don't have too much influence from these other things. So back, you know, with the rock skipping, they didn't test rocks that were, you know, this small and this big. It was within a pretty narrow range that they looked at the effects of size. All right, so Oscar, you were asking a question about my research. Yeah, it took me way too long in graduate school. Yeah, I just had to keep going back, and long story short, I think I just eventually wore them out. I was kind of in the wrong department because what I was doing was more natural resources, wildlife conservation, and management oriented. And at my school, there was a whole separate department in a whole separate college that was devoted to those types of questions. I was in straight biology, and most of the professors I was working with were much more interested in mathematical modeling and genetic analysis and more theoretical stuff. So my research was too applied for their liking, not theoretical enough. So yeah, it just took a lot more time. And the problem is that eventually it might start feeling like you're chewing the same piece of gum every single day. It loses its flavor big time. And what I want to point out too is that it takes a very long time to make any sort of contribution, because, you know, to do some original, why were they being so difficult about it? Because they wanted me to push the envelope. They wanted to show that I did something that had never been done before and taking things in some new direction, and that wasn't really my goal. I was asking some pretty basic questions. You know, what exactly is this lizard eating? Who's eating this lizard? And these were not rocket science type of questions. They were important to me. They were important to the management agencies. You know, how far is are the lizards moving in a day? Are they maintaining home ranges? How many offspring are they having? Some pretty basic questions, but not theoretical, not kind of higher level stuff, right? On Bloom's taxonomy, it was lower level. So I had to modify things. I had to try to go higher in some things. Let me show you bits of a video, and it's okay that we don't have the sound, although it's much cooler with the sound. Inner life of a cell, and there's an 8-minute version with narration. The 3-minute version is more interesting, and I'm going to go full screen to make it bigger, and I can't see what I'm doing anymore, but here we are in the inside of a blood vessel, and we're seeing white blood cells that are not just streaming past. The red blood cells are streaming past. The white blood cells are rolling past, and it's showing the interaction of the proteins that causes them to do that. And so now we're looking at the structure of the cell, and this is the lipid membrane, and it's showing the interaction between, this is the membrane of the white blood cell, this is the blood vessel, and we're looking at how the different proteins interact. What we're going to see is that the blood vessel is signaling to the white blood cell, hey, we've got a problem, we need some help, maybe a bacterial infection or something. So this is the signal that sets off a whole cascade of events. And so then we dip into the cell. Now this is the membrane, so now we've gone into the cell. And I just wanted to point out, so what happened, Harvard they created a whole new animation department just to animate physiology type stuff, because they were learning things that were really difficult to put just in writing. So the shape of all these proteins and how they fit together. This is all very microscopic, so they can't just look into an actual living cell and film this, but they had worked out through X-ray crystallography and fancy techniques the shape of the individual proteins, and so they knew that it could be animated. They wanted to show this, what it would be like if you could travel into the cell. And this part, this filament that self-assembles out of all these individual protein subunits. So one signal makes it elongate, and then a cleavage protein breaks it and makes it fall apart. This is a microtubule, and it's a similar thing where the proteins self-assemble and can fall apart. And I watched about an hour presentation from a PhD student who'd spent about 6 years working on the shape of these proteins, on the shape of the little individual subunits, and figuring out how it is that they could stick together. So that was 6 years of her research, and it was a pretty big deal to show that these could self-assemble and break apart. And it took, a few seconds to show it. So, and that's pretty good, because most people, their research is never going to be shown in any way. But so it wasn't that one, it was the microtubules right here. So here's 6 years of her life's work to show that and to show this. Ta-da! And of course somebody else does the work to show that these motor proteins carry vesicles along the microtubules, and that it was already known that the microtubules are originating out of the centromere. Oh, there's a mitochondrion. Anyway, we'll talk more about cells another time. But I just wanted to give you a sense that yes, a person spends many, many years studying a very specific question, and you're so narrowed down that your insights may never see the light of day, in the sense that people may not understand the bigger picture of how that fits in. So yeah, it is crazy, and that's how it goes. But there are so many scientists now all over the world working on so many different things, and the world's just speeding up and changing very quickly that way. An interesting thing with genetics, well, let's go back to Charles Darwin's time, and we're not going to talk about him much because it's not even the section on biology, but with Charles Darwin, he figured out the theory of evolution, but he didn't know about genetics at all. No idea about chromosomes. And so the big question was, let's say that, well, the big question was, if you have, let's say, a white flower and a red flower, and they mated, wouldn't they then be pink? And if, let's say the red was a new thing, and everybody else was white. So here comes red, it springs up, white, red, now pink, and those offspring are more likely to mate with the white because that's what's around them. So then they become less pink and lighter and lighter and lighter and lighter until everybody's white again. That's what Charles Darwin couldn't answer because he didn't understand genetics. He didn't understand that this trait would persist, that it doesn't get diluted. But in his lifetime, in Charles Darwin's lifetime, Gregor Mendel worked on pea plants, and he figured this out. He couldn't see chromosomes. He didn't know that they were in the nucleus, but he worked out the patterns of inheritance. And it would have been completely the answer to Darwin's question, and it would have really helped Darwin. But either Darwin never saw the paper, which is possible because it was published in German in a small journal, but Darwin did read a lot, and he read in multiple languages, so he might have come across the paper and just not understood it, right? He might have thought, oh yeah, pea breeding, I know, yeah, it's kind of interesting, but hey, I'm into breeding pigeons, and I'm not going to read too much into this pea plants. But anyway, either he didn't see it or he didn't understand the significance of it. And it wasn't until many years later that that work was rediscovered and that the importance of it was understood. And by that time, Gregor Mendel had died, and his work was never known, never appreciated during his lifetime. And I mean, really, it wasn't. The monks, he wanted to teach high school, and he failed his teaching exam, so he ended up being a monk. And he did this research, and it was a lot of work and took many years, and he published this paper, and the scientific world just didn't say anything. Nobody recognized it as being anything significant, and the people at the abbey where he was a monk burned most of his original research, or just, I don't know, used his notes for toilet paper or something. We have hardly anything from him. All we know is that he made this major contribution. Nobody knew anything about it, or recognized the significance of it until, I don't know, several decades after his death. So anyhow, that's how science goes sometimes. So you get a lot of messiness to the data. So they selected, I just chose this one at random, so I don't know the details. It's back to the diabetic patients, but we excluded patients diagnosed with prediabetes, gestational diabetes, type 1 diabetes, or diabetes induced by steroid use or other endocrine diseases, right? So there are different types of diabetes. They wanted very specific. These exclusions enabled us to obtain data from patients with type 2 diabetes. At registration, the attending physician confirmed the diagnosis according to diagnostic criteria, and we included only patients with available baseline physical activity data who did not have diabetes retinopathy. So they don't yet have diabetes retinopathy. We have background data on their physical activity. The ethics committee of the Tenry Hospital approved this study, and written informed consent was obtained from every participant before its initiation. So this is a change in science. Getting consent is really important for human patients, but having an ethics committee review your stuff, there have been a lot of unethical things in science, particularly, Hitler comes to mind during World War II, conducting all sorts of physiological experiments on Jewish people. Throw them in freezing cold water to study how long can a person live in freezing cold water, and you just run the experiment until they die. Obviously, that's very unethical. But the U.S. government also carried out unethical experiments. The Tuskegee trials in Alabama. This was after syphilis had a cure, but the government was interested in studying syphilis more. So poor people in black communities in Alabama who had syphilis were not cured intentionally. They were not even told what the diagnosis was really. Oh, you have bad blood. We'll give you free medical care. Keep coming back. And they just watched the disease progression, as it slowly killed the patients and as it spread to other members of the family and so forth. So plenty of examples of unethical science. So not surprising that they, state outright what the ethical committee was. And this can be a little bit of a problem. It can be a little bit difficult. One of the years that I was doing my horned lizard work, because of how I got the grant for it, how I got some funding, well, anytime it goes through the university, there's an IACUC committee if you're handling vertebrate animals, which I was. And anyway, one summer I hired a field crew. We started collecting data. I went to pay them, and I wasn't able, the university wouldn't issue any checks to these people that I had hired. And I thought, uh-oh, what's the matter? And, I'm out in the desert working on the lizards, and I call up, and the problem was that I had not sought the advice of a veterinarian for how to euthanize a lizard if it was injured. And I thought, oh my goodness, if one of these lizards is injured, I'll just stomp on its head. That's how I'll kill it, if I need to kill it. And but I had to pause everything, go talk to a veterinarian, go look at approved protocols for euthanizing lizards. And oddly enough, smashing its head was an approved humane protocol for euthanizing a lizard. So as soon as I got a veterinarian to sign off on that, if I needed to euthanize an injured lizard, I could smash its head. Then we were able to go get paychecks. Anyhow, weird. I'm not going to go through this study anymore, but it was just an example of kind of how science can be a difficult process. Well, you know what? Let's go ahead and look at some of the data collection. Then they go to outcomes and physical activity, and they're going to have things like tables in there and charts, and they're going to present their results in some way. So you have an introduction, you have materials and methods, you have your results, which are going to be varied depending on the study, and then you have a discussion area where you interpret your results, basically. To the best of our knowledge, this is the first large-scale prospective study to evaluate the influence of physical activity on diabetes retinopathy development in patients with type 2 diabetes. And they talk about, let's see, the data indicated exercise had no adverse effect on the progression and development. In contrast, somebody else said this, however, this association, So they're kind of comparing their results to other results, and comparison contrast type stuff. The present study only included type 2 diabetic patients without DR and revealed the protective associations between physical activity and the consequent risk of newly developed diabetes retinopathy. So what do you know? Being physically active helps prevent you going blind from diabetes. Not a surprise, but you can't just assume that. You can't just say that. If you want to say it in a scientific sense, you have to back it up. You have to go through the process of showing it. So that's challenging. I want to talk a little bit about another challenge in science, and that is that people have biases of all kinds, and you want to identify your own biases and try to work around them or compensate them. You don't want to just, you can't just, maybe you have an idea that physical activity is going to be good for preventing diabetes retinopathy, but you can't just, if you already think that, you have to set it up in a way that you could convince yourself that you were wrong, right? You have to be able to prove yourself wrong so you would kind of in choosing your hypothesis and thinking about it, it's like, how would I know if I'm wrong? You know, what predictions does my hypothesis make? What tests can I do? And what would convince me that I was wrong? And you're kind of trying to prove yourself wrong every step of the way. And part of that is that you can't really prove yourself right too easily. Science doesn't prove that your hypothesis was right. Science says, well, we haven't yet disproven the hypothesis, so we've given support to it. But it sounds very tentative, and it is. And part of it is because things are messy. Data are messy in the real world. Let's look at, again, I know these, it's hard to read, but in this results section, it doesn't really matter what these numbers are for. I just want to look at these numbers here. 0.55 to 1.04, p equals 0.081. And then 0.49, 95% confidence interval, 0.3 to 0.8, p equals 0.004, and so on. So just looking at those data for a second, I wanted to point out the p value. And I'm sorry, I gotta respond to another student from the other section, because people are trying to take their tests. I'm assuming Blackboard's back up. Let me just say Blackboard has been down. If it is up, proceed. And that might happen again. I tried to get everybody signed up before. All right. Man. 95% confidence interval. So for whatever it is, it doesn't matter here, but they came up with an estimate of 0.49 for something with a 95% confidence interval between 0.3 and 0.8. In other words, now let me give an example that makes more sense. In studying the lizards, the Navy funded some of our research because there were lizards on their bombing range. What was their big question? How many horned lizards are out there? Which is a terrible question, because because these lizards like to hide. So if I go out, let's say you go out, you're looking for Texas horned lizards, right? So you stop on the side of the road, you go cross over somebody's fence, because we're in Texas and everything's private, so you're risking your life here. You're walking through the scrubby, thorny bushes, and you're looking for a horned lizard. You search over an hour on a 1 acre area, and in that hour you did not find a horned lizard. Can you say that there are no horned lizards on that acre of land? Good, I'm glad, Oscar, to hear that Blackboard's up. So I posed a question in the chat box here. See a horned lizard during 1 hour searching 1 acre, can you say there are no horned lizards on the land? You know, what are some other possible explanations? Right, you can't, I mean, it's really hard to prove a negative, right? It could take you 30 hours of searching. But if you see a lizard finally after 30 hours of searching, then you've disproven, you know, if your hypothesis, you kind of set your hypotheses up to fail. So you say, there are no horned lizards on this land. As soon as you find one, well, you've proven your hypothesis wrong. You can word it the other way and say, there are horned lizards here. And in this case, if you find one, hey, great, they really are here. So in that sense, it sounds like you're proving it. Hey, you've proven there are horned lizards here. But that's not a very good hypothesis, I'm afraid. It sounds okay. It sounds okay. You're right. They could be hiding. They could be moving away from you. All sorts of possible things there. Right. So you go back to the site again and again and again. Let's say you're trying to estimate density. Okay. You want to know how many per acre on average. So yeah. So let's say you set up a study and you're going to sample every day, an hour here, an hour there, an hour here, 3 different spots. And you go back and you search 1 hour here, 1 hour there, 1 hour here. And you go back every day. And if you find a lizard, you mark it so that you know if you find the same lizard again. So let's say in plot number 2, the first day you caught a lizard, so you marked it. The next day you didn't see that lizard. Well, you don't know if he was there or not. Maybe he moved off the plot. Maybe you just missed him. The third day, you catch your first lizard and you catch a new lizard. Okay, so was that new lizard there all 3 days? You don't know. It might have been off the plot. It might have been there and you didn't see it. So all you can do is estimate. And boy, there are statistical models for doing this. And basically, the more data you get, the better your estimate's going to be. And so this is, you know, I've set up these types of grids and done exactly this type of study. And I can tell you that there are big, big differences based on who's looking for the horned lizard, right? My wife, frankly, is the best in the world for finding flat-tailed horned lizards. And I'm second best. I used to be the best, and then she's very competitive. So she's a machine at tracking horned lizards across the sand. And you know, we just did this for so many years and it's such an obscure lizard, it's not surprising that we became the best. But we would hire new people each year because it's hard to get people to come back a second year to spend, you know, 6 months walking across the sand looking for lizard tracks and picking up, you know, trying to find lizards. Anyhow, and there were times that we'd hire somebody who we thought was good, who just couldn't develop an eye for finding them. And they would go months without seeing one. It's like, oh, this is painful. You know, we're paying you here. You're not finding anything. And other people would get pretty good. So observer bias was huge, just the observer differences. But even with the very best, even with my wife, our estimates came down to, if a lizard was on the plot, she had about a 25 to 40% chance of detecting that it was there. And that was weather dependent. If the wind was blowing so that it would erase the tracks, then that number goes way down. Or if the wind had not blown for several days, then there would be too many tracks. So yeah, the lizards do hear you. The particular lizards we were studying, instead of running away, they would bury under the sand because they lived in a loose sandy area. So we had to follow their tracks to see where the tracks ended, and then you could just reach under the sand and pick them up. If all you were doing was looking at the surface, you would hardly ever find them because they were buried. But what we got, you know, we had to, the mathematical model would take into account who was looking, how many times did you look. We built in, we would give an estimate of the wind for the day and the cloud cover, because that made a difference as to whether we could see the tracks. How low the sun was, what time of day made a big difference. So lots of different factors. The temperature was another factor, and you just have to build these in, and you have to get a whole bunch of data, day after day after day of looking and marking these lizards. And what do you get? You get an estimate. You say, well, you know, there's approximately 2.8 lizards per hectare, or per acre. Well, it was a hectare that we were surveying. And you get a plus or minus. You get a 95% confidence interval. 2.8, but the 95% confidence interval is, you know, between 1.3 and 4.7, something like that. I'm making those numbers up. But in other words, the estimate's here, and you're 95% sure that the real number's someplace in here. And if you're comparing three different plots, plot 1, 2, 3, you're going to have for each one an estimate with error bars, and then over here an estimate with error bars, and over here the third plot, maybe it's down here with error bars. And you wonder, is the population different between here, here, and here? Well, it kind of depends on how spread out they are. You know, there's a difference between here and here, but here to here, that's where you end up with a p-value. So these p numbers here, p equals 0.081, p equals 0.367, p equals 0.009. The p-value is the probability that the things that you're comparing are actually the same. Like there isn't a difference between them. So you want your p-value to be small to say that there really is a difference. So this p equals 0.009, if you times that by 100, you'd get 0.9. So basically there's less than 1% chance that these two things are really the same. So in other words, one plot maybe it was like this and another plot was like that. You know, there's a pretty big difference between them. So there's, you know, less than 1% chance that really the average for both of them was here or something, you know, that really the population wasn't different. Or another way of understanding p-value is, could you get this much difference in your numbers just through random chance? That's what the p-value really means. Can I show you a picture of what those lizards look like? Yes, I can. So here they are. You know, people put them out where you can see them for pictures. There's a picture I took. But there's a close-up. You can see that, you know, it's buried and its head's sticking up just a little bit there. Let me see if I can. Let's try flat-tailed horned lizard. Camouflage, let's try buried. I've got tons of pictures. I guess I could pull them up just on my computer. But, yeah, not finding camouflaged pictures. I guess the point is that you know, when you find the lizard, you take a picture of it where you can see it. And they're not always buried. They are sometimes just out like this. Anyhow, so the p-value, sorry to bore you with p-values here, but what it means is, you know, what are the odds that you could have had these results just by chance and that actually you're wrong, you know? So I compared these two things. They look different to me. I took all this data on them. The statistics help you say, you know, what's the chance that they were really different versus the chance that now they're not different. So the cutoff, and it's just tradition, there's no magic about it, but usually the cutoff is 0.05, like 5% chance. In other words, there's a 5% chance that I'm wrong, that these two things really aren't different, that you know, I just got these data by chance, but that gives you some confidence that you're right. So this one, this 0.081, you're going to say, nah, not statistically different. 0.019, statistically different. And then the 0.009 or 0.004, those are numbers that you're going to have a lot more confidence in and say, oh yeah, there was a real effect here. And so all those different things that we were talking about, like the effects on the rock, the shape, the angle, things like that, it's hard to separate out the influence of all those things, but that's what statistical analysis helps do. It builds a model. You build a model of, you know, the factors for the perfect rock throw or rock skip, and it will tell the relative strength of the different factors, you know, which things contribute the most. And so that's all I'm going to say about statistical analysis, but it is a big important part of doing the biology. I'll show you it's a hard part, and here's a paper that I wrote that, see if I can zoom in on this. The horned lizards would get impaled by a predator, by a bird called a shrike, that would stick its prey onto a sharp stick. And so we collected the dead ones and we measured their horns and heads and everything we could measure on them, and we measured the same things on live ones. This one had a wound on it, but it had survived. And I did statistical analyses to see, you know, what of the measurements we could take, is there anything that would predict whether a lizard would end up impaled onto a stick. And so it took me a long time to figure out what type of statistical analysis to do. I ended up doing something based on a guy who had done work with fish up in Canada, and the effect of different sizes of fins on whether or not they had mates, and the coloration on their belly. Anyway, I found what I thought was kind of the appropriate statistical model, and I analyzed the data, and it came out showing, hey, wow, there's a big strong effect for both the length of the horns in the back and the width of the horns on the side. And it predicts, you know, if you're really small, you got, you know, 20% chance of survival, and if you're really big, basically 100% chance of survival. And with the back horns, about a 40% chance of survival for being small, and it goes up to, you know, about 100% for bigger ones. So the data came out really nicely, and I was like, wow, that's really cool. And it had taken me a long time to carry out this statistical analysis, and I totally didn't believe the results. I thought, I must have done something wrong. I must have done something wrong, because this is just too good. The data come out too cleanly. So I wasn't going to publish the paper at all, and I kind of just did nothing with it. And then my professor said, hey, what did you ever do with that? And I showed him. He's like, oh, this is good. Let me send it to my son, who is really good at statistics, and let him look at it. And he looked at it, and he tweaked the analysis a bit, but he came up with the same basic conclusions. He's like, oh, we got to get this published. So anyway, lessons from there is that, yeah, that's part of collaboration. I just didn't have enough confidence in my math abilities to say, you know, I really did this right. Again, science is about trying to not fool yourself, and I thought that, you know, it was saying, the analysis came out saying exactly what I hoped it would, and so I didn't trust it. I didn't trust the analysis. I didn't trust myself, is what it was. Why did you decide on these lizards? I had a different project in mind for graduate school. I had it all worked out and designed, but it was going to involve living out of my truck and just working on my own in the desert. And just before school started, my advisor, Edmund D. Brody Jr., said, "Hey, I just got a grant to do horned lizards. I don't do horned lizards. Do you want the grant?" I was like, ooh, big grant, funding. It was completely different questions than what I was planning on and everything. But so I just came down to money. I was like, wow, I could actually get paid as a graduate student. So, I mean, not that I got paid a lot, but I got paid. So it was better than living out of my truck, I decided. Okay. So, yeah, science requires a lot more math than I had anticipated. And that's a difficult thing. It is a barrier to entry. It doesn't mean you can still be involved in science without being the one that does the math, and that's part of why teams of people do science, really. Okay, Berkeley has some good resources on science, and I'll put the link here. And I think I posted this to Blackboard as well the other day. It kind of goes all over. There's an Understanding Science 101 and for teachers and resource library. So there's a lot here. How science works, why science matters. Science toolkit is what I wanted to look at a little bit. No, that's not it. I wanted to just look at some of the nuts and bolts of the process, because the process, you can do a lot of teaching the process of science in your classes. So they talk about the scientific method and how that's an oversimplification, that really it doesn't go that way most of the time. So reality, you're just kind of going all over the place and gathering data and testing things and then moving in a new direction and then trying something else. And, so it's really quite messy. So the real process of science here, and they talk about how it's a feedback loop, and you get peer review, you replicate your studies, you discuss with colleagues, you publish. Publishing is a whole other monster. To be quite frank, it's so the ones, Melissa, that you caught growing up were Texas horned lizards. Yeah, so it's a different species that I worked with. But a lot of work's being done on Texas horned lizards right now too. Not predetermined. You don't know where you're going ahead of time with science. And here's an example of not knowing ahead of time where you're going. A guy that I knew studied viruses. He was a virologist, and he studied one type of virus. It's called blue tongue virus. And there's a lot known about the virus now because he spent 30 years studying the darn thing. And it's not known to a lot of people. There's a sheep that's infected with it. The sheep may die from it, but he wasn't particularly interested in sheep. He was really just interested in the virus, and he just chose it because nothing was known about it, and he was curious, and he wanted to learn more about it. And he learned that it had RNA instead of DNA. And I think this might have been the first time that somebody had found a virus that contained RNA instead of DNA. And I do know that people didn't believe him for a while. And so then he figured out that there's this reverse transcriptase, this enzyme, that once the virus was in the sheep, it would transform, it would use the sheep cell to go from RNA to DNA, and then insert itself into the nucleus of the sheep cell, and then start making copies of itself that way. So he wrote about this, and you know, some other virologists probably cared, but you know, a lot of these studies are so boring when you try to write them up that, you know, it would make your own mother cry to try to read them. So he went on for years enjoying his virus and nobody caring about it really. And then the AIDS epidemic happened, and as scientists started looking into AIDS, they found out that it had RNA. And so they did their research. They started looking through publications for, you know, who studied a virus with RNA. Well, this guy had. So suddenly, he's got governments around the world asking him questions and wanting him to study HIV and figure out the virus. And so he gave them information, and he told them everything he could about RNA viruses that he was aware of and reverse transcriptase. But he did not want to go study HIV. He just wanted to continue studying his blue tongue virus. And so his research, you know, there was a big interest in it for a little bit, and then he said, okay, thank you very much. I'm going back to, you know, I don't want interest, kind of. I don't want to be in the spotlight. I just want to study my little virus. So he continued, and I don't know that this ever got published, because it happened at the end of his life. He was trying to grow the viruses in the lab better, and so he ordered some cancer cells. Cancer cells are great because they just keep growing and growing and growing, so they're super good for working on some question in the lab, because you can just keep growing the cancer cells. There's a lady who died in the 1950s from cervical cancer, and we still have her cells all over the world. They're called HeLa cells, H-E-L-A, for Henrietta Lack. And back to ethics, nobody ever asked her if they could culture her cells. They just did. And her family didn't know about it for about 20 years. Anyhow, so he ordered some cancer cells, I think mouse cancer, and he was growing the cancer cells in the lab, and he wanted to see, can I infect them with virus so that I can get more virus? That's all he wanted. He just wanted the source for growing more virus so that he could do more stuff with his viruses. So he infected his cancer cells, or he just put some virus in there, and the next day all the cells were dead. And oh man, I've messed up the whole thing, and so I obviously don't know how to grow cells very well. So he ordered some new cells, and he got, you know, a few petri dishes of them this time, and he tried to inoculate with virus in one of them, and they all died. Can her family make money off that? Probably only in suing the hospital that did it originally or something like that. I mean, they're spread everywhere in the world now, and there's no patent on them. And there are several kind of cell lines. They've mutated so much that there's, you know, this cell line with these weird mutations, and this one with all these other weird mutations. Cancer cells just never know when to stop growing. Most cells, when things go wrong with them, you know, they'll just stop eventually, but that's the thing about cancer cells. So anyhow, every time he tried to put the virus in the cells just died. So he thought, this is curious. So then he got some live mice, and he injected tumor cells into the mice. And sorry about the phone ringing. I assume somebody else will get that. And the tumors grew inside the mice, and then he injected the mouse, not directly in the tumor, but just into the mouse's bloodstream with blue tongue virus. And the next day, the tumors were gone. And so, wow. So this was, like I said, right towards the end of his life. And let me see if this is, yeah, Joseph Lee is the guy that I knew. selective in vitro cytotoxic effect of human cancer cells by blue tongue virus 10. So he got human cancer cells and tried it out, and in vitro means in glass, so you're doing this in a petri dish. Cytotoxic, cyto is cell, toxic is, it's toxic to it. So, he explains that blue tongue viruses infect primarily domestic cattle and wild ruminants, but have never been shown to infect normal human cells. So, humans can't get blue tongue virus. It can't affect normal human cells. But let's see, infection, we investigated with 5 cell lines, including primary human embryo lung fibroblast, which is lung cancer cell, and primary murine embryo fibroblast. Murine embryos fibroblast. That murine is mouse. Human hepatic carcinoma 3B cell line, so a liver cancer cell line, human lung carcinoma cell line, and mouse fibroblast cell line. In this study, comparative analyses of differential cytopathic effects, survival rates using different multiplicities of infection, ultrastructural changes by transmission electron microscopy. We provided the first analytical demonstration and evidence that blue tongue virus 10 could selectively infect and degrade human cancer cells, but not cultured primary normal cells. So in other words, it just kills cancer cells, not normal cells. And they studied the cells, how did it do it? And basically it was convincing the cells to self-destruct, to turn on a self-destruct sequence. So anyhow, I don't know where it's gone from there. He died, so, but other people can pick it up where he left off, and I don't know. So, but just to say, that was just to say that you don't know, there are many routes. You don't know which way you're going to end up going, what it's really going to be about. Okay. So what about with kids in classes? I mean, a lot of it is observation, gathering data, coming up with ideas, just trying to be curious about all kinds of things. So, and, you think about measuring, well, you can measure with a ruler, but, what are other ways that you can measure? And it doesn't have to be exact. I mean, think about, well, let's instead of measuring, let's first think about, let's say kids, whatever age you want to work with, think about things they could classify. Okay, what are some things that in the classroom or at recess or at home, what are a whole bunch of things they could put into groups, because humans really like grouping and classifying. So that's a good, give me some examples. Colors, shapes, and colors is a good example because everybody can say, oh yeah, that's red, that's red, that's red, that's red. When does it become orange? And what's the red orange, or when does the orange become yellow? And you're kind of looking at the transition. How many categories do you want to make? It's kind of interesting too because anthropologists have studied languages and the language of color, and some languages have very few words for color, and the people actually perceive fewer colors. If you say, okay, here are a whole bunch of paint splotches or whatever, put them into groups, some cultures, just from their language, they will just put them into, say, 3 groups, and one group might be red, but blue and green may all be together. And I don't know what, everything that's light in color might be together over here. So, yeah, or you look at cars and all the different ways to classify cars. You're going to do based on shape or size, color, model, who's the manufacturer, the age of the car, is it a truck, is it, so many ways that you could classify. Okay, now measuring is kind of the next step, because measuring is still kind of classifying, or it can be related to classifying, but now you're going to try to put numbers to it, right? So how can you measure color? You can get fancy and use a spectrophotometer. Spectrophotometer. This machine, you put a little sample into it. Let me show you using spectrophotometer. You got to put your sample in a little glass tube. It's usually a liquid or a gas, but you put it in and it will give you a little readout, kind of a graph of the exact wavelengths that were in there. And there will be peaks and valleys at different positions. It might be absorbing in several places. Let's see, spectrophotometer readout or reading. I'm not seeing quite what I was after, but that's a fancy way. You're not going to do that in your classroom, but you're going to be detecting the individual wavelengths that pass through your sample, and so you'll get very specific numbers there, which has all sorts of applications. But you may find another way to measure. You might find a way to measure brightness or intensity, the difference between a pale red and the vibrant red. I don't know, it can be tricky to classify. Now, sometimes measurements are very consistent, right? If you're using a scale to weigh a sample, let's say you have a sensitive scale and you can weigh out 10 M&Ms, okay? And let's say that it gives down to 1/100 of a gram. Okay, so you weigh 10 M&Ms on your little scale and it gives, I don't know, 7.89 grams or something. So you write down 7.89, and then another student comes and they take 10 different M&Ms and they might get 7.93. Well, is it incorrect? No, there's going to be some variation between 10 M&Ms, right? M&Ms aren't exactly perfect. So 7.93, 7.89, maybe another student comes with 10 M&Ms and they get 8.01 or something like that. So you're going to collect several samples and maybe take an average. And again, maybe you're going to have a range of values. Sometimes mistakes are made. Maybe what if one student has a broken M&M, or has 11 instead of 10, or 9 instead of 10. So there's a measurement error that way to try to control it. Okay, so what are some other processes that students can get engaged in? And we have a section on this. Methods. Scientific method. Data collection, experimental design, process skills. Observing, inferring, measuring, communicating, classifying, predicting. Not good. At least from what we have in our little book here. We kind of covered a lot of these things. Inferring, you know, interpreting or explaining one or more observations, often on the basis of prior experience or perceptions. Yeah, making predictions based on things is a good skill for science as well. The communicating, you know, that's where you're showing your data, you're giving a presentation, you're writing something, and of course it's going to be variable. Using, you know, having the kids stand up and do a little video or a presentation or something. All sorts of ways to present. Let me see if I can find this. There is, no, I'm not seeing. I'm not seeing the one I'm after, actually. Huh. Bad search engine. Well, here it is about it. 8-year-olds published scientific bee study. They had some help. But a group of British school children may be the youngest scientists ever to have their work published in a peer-reviewed journal. In a new paper in Biology Letters, 25 8 to 10-year-old children from Blackawton Primary School report that buff-tailed bumblebees can learn to recognize nourishing flowers based on colors and patterns. And they did a great little study, and Bo Lotto is the neuroscientist that I met who helped these kids carry it out. He's at University College London, and he does some pretty high-level stuff. So let me, Bo Lotto bumblebee. He calls his lab the lab misfits. But the thing I wanted to show you is kind of their setup. They have these little tubes where the bumblebees can get a drink, and they inject fluid into the tubes from behind. And the other thing you see is there's a color, and they can change the color. And so they have a whole grid, and they can change. How do you test whether a bumblebee can differentiate between certain colors? You just, you know, certain colors you put sugar water in, and certain colors you don't, and you see if the bees learn to distinguish. You know, you do some training with them. You know, let's say you fill this color up with sugar water and none of the other colors. After the bees have learned this a bit, then you move the position. Is it the position or is it the color? Well, since they can just change the lights on anything in this grid pattern, they just choose 2 new random locations or whatever to be this color, and they put the sugar water in, and they see, do the bees go directly there? So he had this set up already, which is, you know, pretty fancy setup here. There it is. So these can be any color, and the bees just fly in, and these are their flowers that they sample. So this allowed the kids to ask their own questions, design a study, and carry out, you know, real science. And you know, here's a video. Oh, it's going to open up in a window that I don't have. Can't show it to you right now. Anyhow, the paper itself is fun because they broke a lot of traditional rules. So they listed all their names, which is fine, but he wrote the paper with the students' help. So he said, how should we start? They wanted to start once upon a time. So this is a published paper in the science manuscript that started once upon a time. Oh dear. We're going to have to take a slight break. This is a problem with Blackboard. If it freezes, how the test is set, you will get kicked out and it will submit your test for you. So I'm going to pause. You guys can take a little break. Do come back because we go for a long time since we're doing once a week. So I'm going to pause it while I help this other student because her test got messed up. I'll be back within 5 minutes.